Hello, everyone. This is Lianrui Zuo from Johns Hopkins University. Today, I'll present our recent work about the inconsistent MR acquisition in longitudinal volumetric analysis. I'll first investigate the impact and then propose a solution to this inconsistency issue. Magnetic resonance imaging plays an important role in multiple sclerosis, including diagnosis, monitoring disease progression, and also clinical research, such as volumetric measurements and longitudinal study. However, a challenge in MR imaging is the lack of standardization in acquisition. So why do we need consistent acquisition? Consistent acquisition is helpful for radiologists to compare data, enable us to have more meaningful longitudinal data, and also enable us to combine multi-site data to achieve more statistical power. But there are several challenges in consistent MR acquisition, such as the scanner hardware and software differences across different sites. And also some sites, they have like their specific preferences of protocols, and also technicians' expertise and training is also going to affect consistent MR acquisition. CMSC has published several guidelines for standardized MR acquisition, but in our study, we found that there are still variations in MR acquisition, even with those guidelines. And these variations could cause problems in neuroimage analysis. Like in this example, we are showing longitudinal scans of the same subject from baseline all the way to month 24. And in month 12, this subject went to a different scanner, but this acquisition still follows the CMSC guideline. But if we run these longitudinal scans through some automatic algorithms to extract the estimated cortical gray matter volume, we see this big jump in terms of volume versus the acquisition time. And this big jump is likely because of the inconsistent acquisition. And in this study, we dive into this case and provide more insights about the impact of this kind of inconsistent acquisition. So the objectives of this study is number one, we want to investigate the impact of inconsistent MR acquisition in multi-site and longitudinal volumetric analysis. And number two, we want to investigate image harmonization as a potential solution to this consistency issue in MR acquisition. Before we dive into the methodology, I want to spend some time to talk about the data sets. So we use the TREAT-MS trial, which is a multi-site MS trial using approximately 50 sites across US. And this TREAT-MS trial follows the guidelines of CMSC 2018, but this guideline is now mandated. And we acquire longitudinal data of every subject from baseline all the way to month 48. And this study will use a subset of the TREAT-MS trial. Specifically, we have 8 imaging sites, more than 200 subjects, and also more than 500 3D high-res T1 weighted images. Let's see an overview of the study. So out of the 8 sites, we grabbed the largest site, which is the Hopkins site. And this site includes 83 subjects to run our first analysis. So this first analysis kind of forms the best real-world scenario where all the MR data can be acquired consistently from one site. And in this analysis, we first extract volumetric measurements of every subject, and then we fit a linear mixed effect models to these volumetric measurements to study the statistical relationship between cortical gray matter volume and age. And then we run a second analysis. This analysis, we use all the 8 sites, data from all the 8 sites, and then we follow the same image processing steps volumetric measurements, and then linear mixed effect modeling. And by comparing these two analyses, we can kind of have an idea about the impact of inconsistent MR acquisition because even though these 8 sites in general follows the CMSC guideline, there are still some variations between these sites in terms of acquisition. And lastly, we run a third analysis, also including all the sites, but this time before running any image analysis, we first run an image harmonization algorithm. And this image harmonization, not to be confused with statistical harmonization, as you may already heard of, like combat. So this image harmonization, the input and the output, they both MR images. So we want to generate a harmonized image as if this image is acquired using the desired consistent protocol. And with this third analysis, we can have some idea about with harmonization, can we do better for multi-site study? So in the following slides, I'm gonna talk in more details about each image analysis step. So for whole brain parcellation, we use SLUNT. This is an automatic algorithm that takes a T1 image as input, and the output is a parcellated brain into 132 structures. And we can use this SLUNT algorithm to extract some volumetric measurements of our data. For harmonization, we use HACA3, which is a recently developed harmonization methods by me and my colleagues at Hopkins. So the inputs to this HACA3 algorithm is number one, a source site image, and number two, a target site image. So we input those two images into HACA3, and the output is also an image. We call it a harmonized image. And this image will have the same anatomy as the source site image and the same contrast as the target site image. So as if this subject went to the target site to acquire this image. And for the linear mixed effect models, we specifically focused on the statistical relationship between cortical gray matter volume and age. So on the left, we show an illustration of the LME model. On the x-axis is age, on the y-axis is the cortical gray matter volume. And with this LME model, we can parcellate the overall variance of the cortical gray matter volume into two parts. So number one, we call it population variance, and this models the natural variation of the cortical gray matter volume. And the second part of the variation is called residual variance. And this models how noisy our observations are. So ideally, we prefer a lower residual variance for our statistical model. So how do we know how well our statistical model performs? How well we can study this statistical relationship? So we use two measurements. Number one is called longitudinal interclass correlation, or ICC. And by definition, this ICC is defined as the population variance divided by the overall variance, and is ranging from 0 to 1, where 1 models the perfect scenario where all the variances are because of the population, the natural variation, instead of noisy observations. The second measurement we use to evaluate our statistical model is directly from the residual variance. And by definition, we prefer a lower residual variance. So let's see how the three analyses perform in terms of ICC and RV. So in the first scenario, we only focused on one site. And for this one site, we can have kind of very consistent acquisition. This kind of the best real world scenario we can get. but we also suffer from some small sample size because we can only have like controlling acquisition for our site. And in this scenario, we have a almost perfect ICC, which equals 0.96. For the second analysis, we do have larger sample size because we include a lot of sites compared with the first analysis, but we do suffer from inconsistent acquisition. And in this case, we see that the ICC goes from almost perfect to a kind of poor ICC, only 0.65. And according to the literature, this is considered as poor ICC. And on the third analysis, we also include images from all 8 sites, but this time we include HACA3 as a image harmonization step and see if we can have better performance. And in this scenario, we have improved consistency with harmonization, and also we are able to pull multi-site data to achieve larger sample size. And in this case, we can have almost identical ICC compared with the best real-world scenario, analysis number 1. So what about RV, the residual variance controlling how noisy our observations are? We kind of seeing the similar trend. For the best real-world scenario, analysis number 1, we see an RV close to 60. But in the second analysis, when we include all the data, but without doing any harmonization, we see a much worse RV. And in the third analysis, we can kind of have improved RV, and this improved RV is almost identical to the best real-world scenario. So what do we learn from this study? Number 1, we found that even with CMSC guidelines, we still want consistent MR acquisitions. So this consistent MR acquisition gonna produce excellent ICC for our longitudinal study. And on the other hand, inconsistent acquisition gonna produce poor ICC. And number 2, if you really cannot acquire data consistently, it's not the end of the world. Our experiments show that we can use image harmonization as a solution to alleviate this inconsistency issue. And after image harmonization, we can achieve nearly identical ICC as the best real-world scenario where we can acquire data consistently. But before I conclude this presentation, I do want to spend some extra time to talk about image harmonization because we do see a lot of potentials of image harmonization beyond this. So first of all, our image harmonization method, HACA3, can harmonize data beyond T1 weighted images, such as flare images. And this flare is more commonly used in MS research and clinics. On the left figure, we show unharmonized images from 2 sites. We see that they have different image contrast because of the difference in image acquisition. And we then run HACA3 to harmonize those data. And on the right, we see that these 2 harmonized images show much similar image contrast, and also they preserve the anatomical structures of white matter lesions. Second of all, our harmonization method has the capability of taking information from multiple contrasts. And in this example on the left, if we do acquire multiple MR contrasts in a single imaging session, we can kind of utilize this ability to minimize the impact of artifacts. Like this, in this example, the flare images shows really poor image quality. But after harmonization, because our method seeks information from multiple contrasts, we can have an even better image quality in this harmonized flare image. And lastly, our image harmonization method has the ability to quantify acquisition differences in a data-driven way. So in this figure, I'm showing MR images from 5 different sites, and for each site, I'm showing 2 representative images. And on the right, we can project those images into a machine-learned feature space. And we see that images acquired from the same site, they kind of cluster together, and images acquired from different sites, they kind of are separated in this feature space. We found this very interesting to be able to quantify these acquisition differences. And we do also have a CMSC poster that specifically investigates this capability. And lastly, I want to talk about some clinical impact of this harmonization technique and also talk about some future work. So across this study, we found we can improve patient tracking over time using this harmonization because now we are able to handle consistency issues because of like some unexpected changes in patient imaging and also reduce some unnecessary repeat imaging. And also, our studies show that we can improve multi-site trials. This is going to enable MS research to include data with more diverse acquisitions and also enable some scientific findings with improved statistical power by pooling multi-site data. What are the next steps? So number one, we want to experiment with more contrasts like T2 flare, and also we want to compare with more harmonization methods, including the statistical harmonization methods. So before we wrap up, I do want to come back to this opening example. We have already seen that this inconsistent acquisition in month 12 caused us a lot of troubles in longitudinal volumetric analysis. But with harmonization, we can harmonize those longitudinal images and generate harmonized images. And then we rerun this volumetric analysis. We now see a much meaningful and much smoother longitudinal trends of this subject. And finally, I would like to thank all the TreatMS participants and our collaborators from both JHU IACL Lab and also JHU Medical School. If you have questions, feel free to contact me using the following email address and also my Twitter. Thank you.